Hello, everyone. Welcome to this new webinar of the IPPN series. We would like to thank Philip for the invitation, and we're going to speak today about water use efficiency and finding the best trade-off between productivity and survivability. Today with us is myself, Alexis Komor, and Keren Moshilian. Hello, Keren.
Hi, everyone, and thank you for joining us today.
So I am the CEO of Hiphen, and Keren is the CEO of PlantArray, and we're very happy to be here today. So who is Hiphen? Hiphen is a company specialized in plant measurements through a wide variety of sensors, from satellites to drones to phenomobiles to smartphones or to embedded sensors on the field. We're able to measure any type of metrics from plant counts, organs counts, architectural data, etc., on any types of crops. And so we really work in a very various use cases. Today, we're a company based in France with US offices. We work on the 5 continents. So among our main and valuable customers, we can cite Summit, INRAE, University of Queensland, and University of Tokyo. But we also work with the agro-industries, so people like Moët etc., on always the same type of topics, which is how to measure plants for their use cases. And now let's go on to PlantArray.
Thank you, Alexei. So PlantArray is delivering a high-throughput phenotyping solution. The technology was developed at the Hebrew University in Israel by 2 professors, one expert in plant physiology, the other is in soil and water sciences. We are from Israel, and we licensed the technology, and we're commercial since 2017 with already a global presence in 9 different countries. And there are over 30 scientific articles that were published using our solution. Some examples of our customers are Wageningen University and some distinguished customers like Göttingen, Wageningen, Florida, and more. And also on the industry side, we work with our customer base, food and chemical companies like Mars Wrigley, ICL, and others. So our solution is for high-throughput phenotyping indoor in the greenhouse or in growth chambers. And we are performing functional phenotyping, which is physiological screening of the plant on a plant level, plant by plant level, measuring all the plants at once, simultaneously, all along the day in very high resolution. And we perform a quick screening. So within days, up to weeks, you can get the results of your experiment. And this is versus full seasons in the field. And we do 3 main things, all in parallel, all automatic, control the soil and water of the plants, measure the plants physiologically and also environmental analysis, together with analyzing it in real time, including statistics. And the outcome is that we predict the plant performance with high correlation to yield results in the field.
Thank you, Karen. So today's talk is about some ideas on how to monitor water use efficiency in a quite efficient way at the same time in the field and the greenhouses. So we're going to start by speaking a little bit about the issues and what's at stake for the water use efficiency. Then we will focus on how we can go from the greenhouse to the field or from the field to the greenhouses. And then we will go into more details on how we can assess this type of measurement at the same time in the field and in the greenhouses. So let's get on with it. So why water use efficiency is such a big issue today? Well, mainly because drought is really a topic that is getting more and more importance due to global warming, due to irrigation issues, where we're having more and more crops that are irrigated with less and less water resources to be able to irrigate them. And so all the game nowadays is how are we going to be able to maintain our yields with less water and more extreme environments to perform those high yields that we're doing today.
So it takes today a long time to breed for drought tolerance. It's a very long process, 20 to 25 years. And we also see that there are few results in the field, few solutions, few products out there. And also research, just research on the topics also take a lot of time. And one of the main bottlenecks for this is really the ability to measure the crop physiological, complex traits, physiological traits such as water efficiency and other functional traits, and then correlate them to genetic information, genetic data. Most of this is because physiological traits are measured mainly today still manually or by plant, and so it's a very slow process.
So in order to go into the definitions a little bit, and of course definitions is always something that can be discussed, and different teams can make them vary a little bit. But for us, what is water use efficiency is the amount of water needed to make yield, to make a quantity of yield. So then yield can be divided into biomass and harvest index. Water use can be divided in evaporation, transpiration, and losses you're going to have. But basically, that's all what we're going to discuss about is how do you use the water in order to achieve your yield. And for that, there's many processes at stake in the plant that are going to control and to improve the quantity of water you're going to use to achieve your yield. It can be either the stomatal conductance, but also the capacity of the roots to extract water, the quantity of fruits, etc. So there's many processes involved in this water use efficiency, and that's really what we're going today to focus on, how we can try to disentangle all those processes to find the most efficient plants.
Looking at water use efficiency alone is also not enough. In order to learn the ability of the plants to be resistant to drought, we need also to evaluate the productivity of the plant versus its survivability. And let me explain what I mean by that. You can see that here are two examples. One is with the sunflower and the other with the cactuses. So sunflower is just an example for breaded field crops, and cactuses for those plants that are living in harsh environments like with low water amounts. And what we can see, of course, the sunflower and such field crops were designed to produce more yield. And this is what they do. They produce more yield. When we look at their stomata activity, we can see that their stomata is mostly open. They produce and they do a lot of photosynthesis, and transpiration is also very high. So they lose a lot of water. So they're also taking a risk. What will happen tomorrow when there is no rain? And how will they cope with that when they lose a lot of water? On the other hand, we have those plants that are living in harsh environments. They developed a mechanism where their water use efficiency is very high, but the production is low. So photosynthesis is low, their stomata is more closed. They're not losing a lot of water, not doing a lot of transpiration, but water use efficiency is high, but production is low. So what we would like to do in the, and what is better to try to achieve in the process of finding tolerant plants to drought is really to find a balance between the two. Water use efficiency, but also look at this balance between productivity and survivability. And we'll explain how we do that in the next session.
Thank you, Ken. So now let's focus on how visions we're having to go from field to greenhouses in regards really to this water use efficiency applications with Hiphen and PlantDetect tools. So the first thing we would like to mention that it seems to me really important is scalability. So the ability to measure a lot of plants, especially in nurseries, can nowadays be achieved in the field. And this is something that is really new and massive with digital tools. On the other hand, being able at the plant level to measure through PlantArray system, by example, the processes of a wide variety of plants is also arriving to maturity. So that's why we do believe that there can be combination in two sides. Karen?
So the way we propose in this presentation is that you can go from the lab, from the genetics, towards screening, doing an efficient screening of physiological complex traits in the greenhouse, measuring on a plant level, and trying to find to do a simulation, what is the best performing plant based on physiological data. From that, going to the field and screening for the best plants, and of course matching it with genetic data. Or do a reverse phenomics where you go from the field, you try with large quantities of plants, with large populations, and identifying those that have the potential, going back through the greenhouse to understand it on a plant level and to the genetic side. Link it to the genetic side, in both cases, of course. So we offer here two ways of approach, which makes the whole process very efficient. And we will present to you in the coming slides how we deal with the water use efficiency and other functional traits in the greenhouse and also in the field in both ways.
Thank you, Ken. So let's go into how we now concretely evaluate that in the field. And really, I love this idea of being able to do two ways, and the digital tools are really allowing us to envision new ways of making selections in this field. So first, what I think is important to mention is nowadays we arrived at the maturity to be able to evaluate agroclimatic traits. What are agroclimatic traits? Agroclimatic traits are traits that combines at the same time plant data recorded by drones, phenomobiles, or other sensors that take data in the field with climatic data like weather stations and soil data, soil probes, etc. And mixing them together allows you to get agroclimatic traits. I guess the most important one is what we call a QPAR, or the quantity of light intercepted through the season, or the energy that is intercepted for the light use efficiency. We can also mention plant phenology, so to be able to understand when each phenological stage arrives, and of course, water use efficiency. So now on, we're going to focus on concretely how do we manage to mix those data together to be able to have some insights on water use efficiency. Well, I think it's really about an evapotranspiration assessment. So basically, we're able to have an F cover, so a fraction of vegetation on your microplots. By combining it with a water balance model and some consideration about your water availability in the soil, evapotranspiration and transpiration processes, you're able to extract traits about water stress. So let's get a glimpse about this water balance model, how it works. Well, it's quite easy actually. Yeah? It's really about considering that you're having some water availability on your soil, that some of this water is going to go for evaporation, some will be able to be used by the plant for transpiration. This transpiration can be at its maximal potential as long as you have a certain amount of water, then it will lower down because there's not enough water to extract, so you will still have an evaporation, but a transpiration, but less. And finally, you arrive to this wilt point where the plant dies. And so all the game then is to say, okay, how much does it rain? how much does my plant transpirate, how much does my soil evaporate, what are my water loss, and I'm able to seal that. For evaporation and transpiration, we're using really those Monteith equations that are, I guess, quite standard in our field, that allows us, by comparing with what would happen with grass, to evaluate the ETP, so the potential evapotranspiration, and we're able to transform the ETP into ETR through the water balance model, and we can discuss that further if you want. And that's really the idea on how, with just some simple drone data and climatic data, we're able to really go much further and give you some information that are comparable across sites, and that are really easier to handle it in a massive big data scale. Some concrete examples. Here you can see the example of a plot that had actually a very strong vigor, a very strong initial vigor. This very strong initial vigor meant that you had an F cover that developed fast. So you had some the ETP versus the potential transpiration. So the potential ETP versus the real ETP, and especially on transpiration, was soon starting to split, and you had water stress arriving extremely rapidly. On the other side, this microplot, which had much less vigor, so where the F cover started increasing much later in the season, meant that your transpiration rate were lower, so you match your ETP for much longer, and stresses occurred later in the seasons. So you can really see the difference between those two sites. So that's the theory of this, the way we are managing to compute water stress. Of course, and that's really, really important, that this model tells you that we should have had water stress or not, doesn't mean that the plant stressed, because there's many other processes at play, like the quantity of water availability in the soil, the routes to extract this water availability, the stomatal conditions that can be a little bit different, and that's exactly why you're breeding for those things, which means that actually we need to disentangle, was this stress a real stress, or was the plant able to, it should have theoretical stress, but it didn't, and so it's a plant we want to target. For that, we can do canopy temperature assessment, so to look at if the plant is heating or not. If it's heating, it means transpiration is lower because the stomata are closing, and thus that it's starting to stress, or at least that the potential evapotranspiration is not rich, and that's so your real transpiration is much lower, which means that yes, it's stressing. You can also look at leaf rolling. So leaf rolling is also another trait that is instantaneous, which is about limiting the F cover, so limiting the surface of transpiration in order to use less water and to be able to increase survivability. There's many other processes we could think about, typically fluorescence or other processes, which we don't yet measure, so but we're working hard on. And then the first step is to say, okay, now that I have those agroclimatic traits, I know my stress and all that, I look actually at what yield I achieve compared to the stresses, to those agroclimatic traits, to look at efficiencies. And there is really those plants that has very strong, even if the plant doesn't have outstanding yield, if they have outstanding efficiencies, I do believe it's those plants you want to select, and you want to understand why they're performing so well. So let's go just for a couple of minutes into those examples of instantaneous stress that I really think can complement those water stress assessments we presented to you. The first one is temperature. So temperature is a really, really tricky trait to play with, because soil is always much warmer than the canopy. So if you look at the global temperature of your canopy, you will be sensitive actually to the F cover. So lower is your F cover, higher will be your temperature. Higher is your F cover, lower will be your temperature. whatever is the stomatal conductance. So we're working hard on trying to disentangle the soil to the soil's effect from the plant heating up. There's already some papers discussing about that, and I believe that the solutions will come with cameras with higher resolutions. The other point which I contributed for this publication is how can you assess leaf rolling through time. So today there's some digital ways that are as good as manual and visual scoring, and I'm really happy to discuss that in another webinar with you in details. And so once we are managing to have all that, is a moment where we say, okay, we did all this work, why not in nurseries and tens of thousands of plots in a really digital way where we use big data, and I managed to find a certain number of cultivars of modalities that are outstanding. What do I do with it? Well, probably the first step will be to validate that it's really outstanding cultivars. Why not with PlantArray systems? And I give the hand to you, Karen.
Thank you, Alexei. So yeah, let's discuss how we validate this in the greenhouse. This is the PlantArray system. What we can see here is an experiment in the greenhouse on a single plant level, where there is a drought experiment. So the experiment goes automatically from the well-irrigated stage to drought and recovery. What you can see in the graph is a transpiration, daily transpiration parameter, one of the parameters we are measuring. I will speak in a second about all the different ones. What we do in this analysis, we do physiological tests. We retrieve physiological data from all the plants simultaneously, and also on a continuous basis throughout the day, while we control different soil and water cocktails or conditions to each of the plants. So you can provide different levels of water, nutrients, salinity, a combination of those in your research. The system is working on the basis, the principle of water movement and water measurement from the root activity of water influx through the water balance within the plant, and then the transpiration to the atmosphere. So we are measuring the water use efficiency, but not only, we are also measuring the growth and productivity by the plant, because productivity is measured by the transpiration rate. Transpiration is well known as having a direct link to photosynthesis. So as much as transpiration rate is going up, we will see also photosynthesis going up, photosynthesis activity, and therefore yield going up. In this cartoon, we can see in an illustration how the system works. So supplying different soil and water quantities, cocktails to each individual plant. And on the right hand side, you can see all the different parameters. So let's start with transpiration. Precise transpiration measured by the system. Biomass, precise biomass gain is measured by the system. Water use efficiency, which is a derivative of transpiration by the biomass. And root influx together with stomatal conductance. We will talk more, and I will provide more examples. These physiological traits are measured together with the environment, and some of them has very valuable relation to drought testing. All of this can be done for different type of plants. We worked with many types so far, and from Arabidopsis level to even up to trees. I didn't mention, but all this data goes into a cloud software, which then it is being analyzed over there. So what we see here in this example, it's just an example of the different traits: biomass, transpiration, stomatal conductance, drought resistance. We talked about the how it's tough to do to organize the genetic ranking when you do it, the measurement on a plant by plant basis. And here it's just a quick display, even during the experiment, of the biomass, transpiration, stomatal conductance, and all that. This is on tomatoes. Let's go to the next example. This is on tobacco. You can see on the left hand side, transpiration rate, which is being measured on a daily pattern. You can see the daily pattern and the difference between 4 different genotypes. And you can see that if you don't measure the plants on a continual basis, actually you might lose some very important data about the behavior and the response of the plant. In this case, this gap between the upper genotype and the lower one at noontime accumulated on a day-by-day basis to 15% more yield. This was published, you can read the paper. On the right-hand side, you can see another parameter is the biomass gain, how it's demonstrated, again in tomato, and the biomass gain measured during well irrigation and during drought. In this example, we now will focus more on about the water use efficiency trait. This is taken from a paper published by Wageningen University. This is the setup at Wageningen. You can see on the left-hand side a daily pattern of 2 consecutive days. This is the light behavior. At the middle graph, you see how the transpiration is behaving, the transpiration rate, and you can see that it kind of follows the same pattern as we saw on the light, very similar pattern. On the right-hand side, you see the water use efficiency, again, following very similar pattern, but what you can see here, that, for example, this red cultivar, this red variety that was at the bottom with the low transpiration rate actually becomes high rated in the water use efficiency. So the system is measuring all of this instantly during the experiment. Now, we said that it's not enough to measure water use efficiency. We have to look also at productivity versus survivability. This is very important. The trait which is helping us to do that is the stomatal conductance. It is actually measuring this transpiration rate normalized to the plant biomass and to VPD. VPD is the vapor pressure deficit. This parameter was also tested in different cases. It's also published. This is taken from the Wageningen paper where it has a very great fit, 0.99 to the porometer measurement. Let's look at the right-hand side graph. What we can see in the graph, we can see the radiation, the line in red, and then we see in green the VPD. We can see that VPD is low during the morning, and during noontime it's getting higher, which is not surprising. And then we see the stomatal conductance behavior in blue. What we see is that there is a certain peak, a very high peak during the day. And this is because in this time of the day, the plant is most efficient, is producing much more. The production is high, but is losing a low amount of water. This is the most productive time. This is the balance between the productivity and survivability we discussed before. Now, of course, you can measure that with the system for each of the plants on a plant basis, and you can compare between the plants and see who is having this trait in the most high rate, and compare between all the different plants. We tested the system in the greenhouse and with the same cultivars also in the field. We see very high correlation between the greenhouse and the field. You can see the graphs here in well irrigation and in stress. You can see the same behavior and very high rate of correlation. So in summary, with the PlantArray system, you can measure in the greenhouse or in the growth chamber the water use efficiency activity and also the stomatal conductance activity compared to all of the plants simultaneously and continuously. And what you can do is either screen in the greenhouse for a simulation and then go to the field with the best plants, or in a reverse way, do the trial first in the field, and go into more, after you have the discovery, into more thorough study in the greenhouse.
Thank you, Karen. So I guess really the takeaway home message here is that we're really arriving at maturity to have professional digital tools to accelerate drought research. And that's really important because today we still have the sensation that it's researchers that are making amazing research on one specific topic, and that the tools, the overall tools at the same time in the greenhouses and in the field were still a little bit lacking or lacking of maturity, let's say. Today, companies like PlantArray and Hiphen, and of course a lot of others, are starting really to have some scalable tools for you to make your research in a real professional way. And we're really happy to discuss that further with you and to see how we can help you scale your project, scale your breeding programs in order to lower the time where you can have products into the field to the farmers. Do you have any questions? Because Karen and I would be really happy to discuss further about all what we discussed up to now. Please don't forget, we're here to help, and you can reach us whenever you want.